Correct. The DNA is the template. The RNA is like the copy, the transcript, you could say, that leaves the nucleus.
And you said the DNA can stay, the DNA has to stay inside the nucleus, but the RNA could go out.
Exactly.
So it kind
Perfect.
of works like a coenzyme for the RNA. No, it transfers information.
Yeah, exactly. Yeah. Yeah, it's like, so the names transcription and translation are actually kind of, for once, good descriptions of biological terms. I feel like we always get tripped up with names that aren't really descriptive. But transcription is like the copy of making a transcript or a copy. So we make a copy of the DNA that can leave the nucleus, and it's going to be in the form of RNA. Because if we made the transcripts in the form of DNA, what could happen?
They could get confused.
It's true. Yeah, they could get confused and they wouldn't leave the nucleus. There'd be a lot of problems. If we had copies of our DNA, every time we wanted to make a protein, we made new DNA, then the cell wouldn't know like what's new, what's old.
I wonder what the helix get confused too, like would it hop on the RNA and not hop on the DNA?
Yeah, so if transcription made more DNA, then a lot of the enzymes would get confused and they would do the wrong thing because we'd have multiple copies of the DNA out there and it would cause lots of problems. So that's why we have the RNA, which is a different molecule, different type of molecule, different type of nucleotide with all the information that we need to make the protein. So it's, what's that? It would be very, yeah, you're right. So RNA is similar to DNA, but there's two main differences. There's a different type of sugar. So DNA has a sugar called deoxyribose. RNA has a sugar called ribose. So that's where the D and the R come from in DNA and RNA. So a different type of sugar. And that kind of lets all the other biomolecules and proteins know what sort of nucleotide it's dealing with, because it can sort of see that sugar and it can know as it attaches either, okay, this is a DNA or okay, that's an RNA. And it helps the whole cell mediate this whole process. The other difference is there's a different base in RNA that's not found in DNA. So in DNA we had A, G, T, and C. In RNA we have A, G, U, and C. So we have no more thymine, but we have a uracil now. So it's just a different, one different base. So those are the main differences. Oh, also, sorry, the DNA is most stable when it's double-stranded. So it's really going to always be found in like a double-stranded configuration unless the helicase comes through and splits it open. RNA, on the other hand, is often found in a single-stranded configuration. So we make one copy of one strand of DNA, and that's a molecule of RNA, and it just exists as a single strand. It doesn't form a helix. It doesn't form the two strands.
So with the A here with the U, the absence of the T.
Right, correct. Great, good point. Those are the main differences between DNA and RNA. And again, DNA does not leave the nucleus, RNA can leave the nucleus. So what we're doing in transcription is we're going to have a template strand like we did during replication. So we're going to have a strand of the DNA that we're going to be reading. And we're going to have a polymerase, again, that's going to come in and do that. But it's not going to be a DNA polymerase, it's going to be an RNA polymerase because it's going to be adding RNA bases. And as it adds these bases, it's not going to stay attached to the DNA. It's just going to kind of like fall off the chain and let DNA close behind it. Because the bond between DNA and RNA is not very strong. It's not like DNA and DNA that kind of snaps together and stays bonded unless we have something that breaks it up. The RNA and DNA bond is very weak. So after we add that base, that RNA base, it just kind of falls off the chain and allows the DNA to close behind it. So the RNA polymerase also only goes from the 5' to the 3' end. And it's going to attach to what we call a promoter. And the promoter is the region, the first part of the DNA that we want to start copying. So within this DNA, we have, obviously we have this really huge stretch of DNA. And so the polymerase is only going to bind to very specific parts of the DNA called the promoter region to start transcribing. And that's going to be like the beginning of the sequence that it wants to read. So the DNA, again, has information on how to make proteins, and the proteins are going to be read from very specific regions on the DNA, beginning at these promoter regions. That's where the polymerase is going to attach in the beginning, and it's going to go along the DNA, and it's going to transcribe the DNA as it goes.
I have a question.
Yeah.
And you may not know. So some viruses are double-stranded DNA viruses, and some are just regular, not double-strand.
Yeah, I wish I think some viruses. What is it? Oh, so a lot of viruses, like, people technically consider not even to be living because they require like another cell to be functioning. I think some viruses don't have DNA. Maybe that's not true. I don't know. Maybe I should look this up before next class.
No, it just
Yeah, viruses are really interesting. The way so what a lot of viruses do is they'll get into your cell or into a cell and use the existing cellular machinery that's there to replicate themselves and grow. So viruses usually don't replicate on their own. They have to be like infecting another cell, and then they can replicate themselves and they can divide and grow.
Like making evil clones.
Exactly, like making evil clones. That's basically how HIV works. It just makes evil clones of itself.
And that's a double-stranded DNA virus, right?
I don't really know. I'm sorry.
That's okay.
Yeah. But what's interesting, one of the reasons why why HIV is so hard to treat is because it has a very high mutation rate. What that means is it's constantly changing. So the way the polymerase works there, it doesn't make very exact versions of itself each time it replicates. It has a lot of changes as it replicates. And that means that it's a very genetically diverse virus. There's many different kind of copies of it that can respond differently to different drugs or different chemical environments, and so it makes it very hard to treat because you can kill 95% of the virus, but the 5% that develop these mutations might be resistant to whatever drug you're using. Yeah.
And then if those 5% are still there, they just keep running.
And they keep mutating too. So it's not just that you can use two, because two drugs in it. So that's why we use what are called like cocktails of drugs. So it's like a mix of drugs that could affect lots of different. Cancer does something similar. Cancer has a very high mutation rate as it copies its DNA, which makes it very genetically diverse, which is good for the tumor and bad for us. Okay, so the steps for transcription. So we have initiation, elongation, and termination. So these are straightforward names again. We're in luck. So we have RNA polymerase binding to the promoter. That's initiation. We're beginning this transcription process. I thought you were really angry and going to come attack me. We have initiation. The RNA polymerase is binding to the promoter. That's the beginning of this transcription process. We have elongation. That's where we're going to be making a longer and longer strand of this RNA molecule as the polymerase goes along the template strand of DNA. And then we're going to have termination, where the RNA polymerase is just going to hop off the DNA strand. And we'll just have a separate strand of RNA. So those are the three steps. We're going to go into a little more detail on each one. Oh, sorry. But before we do that, we're going to talk a little bit about how what happens to this mRNA after it's copied. So we have some processing that gets done. So right after we transcribe this RNA, we have what's called an mRNA transcript. So the M stands for messenger. This is called like messenger RNA. So this is RNA that is basically like carrying a message from the nucleus out to the cytoplasm. So we have this mRNA transcript that gets processed a little bit further before it's used to make a protein. So we're going to add a cap to the front of the mRNA called a 5' cap that you see right here, and that'll tell the rest of the cell where the front of the mRNA is, so it knows how to transcribe it properly. So this kind of gives it some directionality. On the 3' end, we have what's called a poly A tail. And the poly A tail is going to prevent degradation. So this is just like a long tail of RNA bases that don't contain any information. Thanks very much. But basically, they're going to prevent the RNA from being degraded by enzymes that will just attach to the end and kind of go up the mRNA. So right when this goes out to the cytoplasm, there's going to be some enzymes that start attaching to it and like eating away this poly A tail. And we added that tail there to prevent it from degrading too early. But we do want it to degrade eventually, because that's sort of the way the cell regulates this protein generation process. It makes these mRNAs that go out to the cytoplasm, and after a long enough time, they kind of like self-destruct. They get eaten up by enzymes in the cytoplasm. And that means that we can make an RNA, an mRNA, and send it to the cytoplasm, and it'll get translated into protein a few times. And after a certain amount of time, it just gets degraded, and it no longer makes any more protein. So it makes it like a temporary signal. Like, so we'll just make some of the protein when we make the RNA, but it won't just keep making it forever. Because if it did, then our cells would always have like kind of the same proteins, and they wouldn't change with time and change in relation to our environment and things like that. We also have what are called introns and exons. So there's parts of this mRNA transcript that we're going to use to make proteins, and parts that we won't use to make proteins. So the parts that we cut out and we don't use, those are called introns. Maybe not the most convenient name, but you can think of introns as like in the way. They're in the way, they shouldn't be there. The parts that we will use to make the protein is called the exon, and that's like what gets expressed. Exon is like expressed. So we have this mRNA transcript up there. We're going to add a 5' cap and a 3' tail. We're going to cut out these introns that are in the way, and at the end we'll have our final copy of the mRNA. It's a cap, only exons, and the tail at the end. So now the cell knows which way it's facing from the 5' cap. It knows it won't get degraded too soon because it has this tail, and it only has the RNA that it wants to express, the exons. So why do you think the cell has these introns and exons? That just seems like more work. Why would it do that? Any guesses? Just to confuse students?
So it's consistent and precise in the process.
That's so. How would that make it more precise, though? Well,
Product.
what if it just didn't have them at all? Wouldn't it still be consistent?
I don't think so.
But it helps with the error rate.
Yeah.
Why does it help with the error rate?
Like, it helps to make sure, I would say for like the proofing part, that it keeps that percentage of 1 per 100,000 that will be correct versus the ones that would be, I would say, irregular. Right.
So you're saying like maybe there's more mistakes in the introns that it's cutting out, and so it leaves the exons with no
Either
mistakes.
mistakes, parts that may have been damaged, maybe not.
That's a good guess.
Are non-coding introns getting removed because it delays the next phase that the mRNA goes through?
Sorry, say that one more time.
The introns, the ones for like non-coding introns, they have to be removed because it doesn't allow the DNA to move on to the next step in the process.
Right. This is part of the RNA though, not the
RNA.
These are all good guesses. Maybe I haven't given you enough background information. Let me give you more hints. So, yeah. So, one of the reasons that the cell does this is it allows it to take one section of the DNA and make lots of different proteins from that same section because it cuts out different regions at different times. This is called alternative mRNA splicing. So it cuts out different introns from the mRNA transcript when it's making different proteins. So if this was the initial transcript up on the top, it can cut out different regions. Like here it cut out the green region, and here it cut out the yellow region. And so the final protein that it makes at the end is going to be different because it cut out different regions. And so this allows us to have more information from the same amount of DNA because we cut it in different ways. We edit it in different ways. And this allows the cell to be a little bit more complex because it, for some reason, wasn't complex enough already. But as it gets more complex, it can actually respond in different ways to different chemical environments and different external stimuli. And that makes it more flexible and it makes it more likely to survive, and it can do more kind of more complex tasks.
Oh, it's the big changes in the journal.
Yeah, yeah, it just makes us, allows us to make different proteins from the same stretch of DNA.
So during our alternative mRNA splicing, it's cutting out certain parts of RNA?
Exactly.
Okay.
Yeah, but it cuts out different parts at different
So it
times.
works like checkpoints, sort of, kind of too.
Yeah, sort of. Like, yeah, so it the splicing is like a type of checkpoint, and it looks to see like, okay, what are we doing again? And then it cuts it, it edits it in different ways. Like if you had the same idea but you're presenting it to different people, you would kind of like use different terminology. It's sort of a similar idea. It has the same idea in mRNA transcript, but there's like slightly different needs, different audiences, so it'll make different types of proteins. Okay, so just to review, what was the objective of transcription? Why are we doing this?
To make RNA?
Yeah, to make. Why are we making RNA? Anyone else? It's almost the weekend. Please participate. Why do we? Yeah, great. Yeah, great. So we make. We have a nucleus with our DNA that doesn't leave. That's a excellent DNA. And the DNA like cannot leave this nucleus. So what we can make though is we can make mRNA copies from the DNA that can leave the nucleus. So we're undergoing transcription to make copies of the DNA that can leave the nucleus. And so where does transcription occur? In the nucleus. Where do you think translation is going to occur? In the cytoplasm. Yeah. Sorry, what did you say? Oh yeah, that's right as well. But they're in the cytoplasm. So what enzymes were involved in transcription? What was that? RNA polymerase. So RNA polymerase is like DNA polymerase, but instead of laying down DNA, it lays down the RNA. But why doesn't the RNA then just stay binded to the DNA and we have like a big mess? Right, we don't want a big mess. But physically, why doesn't it stay binded to the DNA? It's the RNA DNA bond. Where's my other marker?
Is it just temporarily there?
Yeah, exactly.
Hydrogen bond or not? It's covalent bond.
No, it is a hydrogen bond. I'm sure. You should always be skeptical. So this is our polymerase. And it's gotten onto this DNA and it's making copies. And then after it makes this copy, our RNA just comes right off because this bond is not very strong. It's strong enough to keep it there for a little bit, but just with the vibrations from the thermal energy in the cell, it's going to kind of fall off and just stay disattached, which is good because that allows it allows the DNA to close in and go back to the way it was. No, it's just because the nucleotide is like a different shape. It just doesn't fit in as well. So but then that leads it to being single-stranded. So it's kind of like the opposite. Yeah. So why didn't we just send DNA out of the cell to make the proteins? Why do we need this RNA nonsense?
It can't leave the nucleus.
Yeah, but exactly. So DNA can't leave the nucleus. This is so bad. I don't know what happened here. I have to try one more time.
Produce protein.
Is that better? Oh gosh. But we can't send the DNA out of the cell because then our DNA will get damaged. And then as we undergo more and more cell divisions, more cell cycles, we would have different DNA each time, because when you're making our proteins, we're sending it out there and it's getting damaged and degraded. So instead we make this RNA that leaves the nucleus, and we keep our DNA safe in the nucleus.
RNA not get damaged?
RNA does get damaged, but we don't use it to replicate. So those damages don't get passed on to the next generation.
Did DNA get damaged during like phase?
So DNA can get damaged when you get a sunburn from like different chemicals, from radiation, and those damages to our DNA can lead to like cancer. So it can get damaged, and it does get damaged frequently, but it usually gets fixed, or it's small mistakes that don't really accumulate, or things like that.
Oh, okay. So does RNA repair itself?
No, RNA is just sort of not as valuable. So when we undergo the cell cycle, there's no copying all of our RNA. There's only copying our DNA. So the DNA is the only thing that gets passed on from generation to generation. And so when you think about it that way, you can kind of think, okay, DNA just holds information. But DNA also has a chemical structure that directs different types of RNA to bind to it. And the chemical structure of the RNA is then going to get read by the ribosome and make different proteins that are going to have a different chemical structure that do different functions. So there's like kind of this intertwine of like information and like structure at the same time. So DNA carries information, but it does that through its structure.
So DNA is the main control center. It's like the brain of a person. You say pick your arm up, you pick your arm up.
Yeah.
It's directing RNA what to do.
But there's no like conscious being in the cell. This is all just like a series of chemical reactions that happens in a really predictable manner, because we have certain enzymes and we have a very specific chemical environment that the cell maintains. And so this kind of all happens. I think it's like the really cool thing is this hugely complex process that just happens on its own very reliably and allows like complex life to exist.
So does DNA makes the RNA? Not really.
Well, the enzymes make the RNA from the DNA. So there's this RNA polymerase comes in, an enzyme comes into the nucleus, it goes to the DNA that it wants to go to, binding to the promoter region. It copies the DNA, makes a transcript called this mRNA transcript. That transcript gets processed because we add the 5' cap and the 3' tail, and we do our alternative mRNA splicing. We send our final transcript out of this nucleus into the cytoplasm, and then next class we'll talk about how we read that mRNA to make proteins.
So when the RNA comes into the nucleus, what's on it? Is it blank?
When it comes into the nucleus, it's just a bunch of individual nucleotides. Okay. So that's a good question though. Let me. Constantly floating around the DNA, we have individual. Oh, that's not a great place to draw that. But just like floating around, we have all these individual nucleotides, and they're going to collide with the polymerase, and then they're going to get matched to the nucleotide. I guess it'll be over here. Get matched to the nucleotides of the DNA and bind along a chain to make one mRNA transcript.
So I guess what I'm trying to say is, like, you know how on your picture you have like a line, but just to the left side, this left one, like all the other, yeah.
Oh yeah.
The other stuff, yeah, right there. So you have like a strand, and then you have like little lines coming off of it. Yeah. So each one of those is like nucleotides.
Yeah.
So when the RNA comes inside the nucleus, is it just that line without the nucleotides, or?
No. So it's these. This is the backbone. Like this is one nucleotide. This is a nucleotide.
Okay.
Like it's this whole. This is the sugar and the phosphate, and this is the base. Yeah. All right. See, we can end early. We did it. Good job, guys. Have a great week.